Welcome to this CAMAG webinar about comprehensive fingerprinting of botanicals. I'm so glad you could join us today. I'm Stefan, CAMAG's Head of Sales and Marketing. A lot has been going on in the world of high-performance thin layer chromatography, or as we call it, HPTLC. The technology is entering a new era with full automation, and we constantly improve our methods to meet the criteria of the modern routine quality control laboratory. But first, for those of you who are no HPTLC experts yet, let me quickly summarize why people are using HPTLC. HPTLC allows separating any matrix with minimal sample preparation. Therefore, it is a technology of choice for samples derived from natural origin, because they often contain complex matrices. The separation is performed with a liquid mobile phase on a glass plate covered with stationary phase. It's not just silica gel. The separated substances are visible on the plate or can be detected by derivatization with chemical reagents or by scanning densitometry. This allows you to perform multiple detections of your target analytes directly on the plate. This workflow leads to reliable and quantitative results. As the stationary phase is only used once, there is no cross-contamination. So today we will learn more about the analysis of Ginkgo from the field of botanicals. And I give the word to Melanie.
Welcome to our webinar about comprehensive HPTLC fingerprinting. My name is Melanie. I'm working at CAMAG as Scientific Business Development Manager and editor of our CBS journal. Besides this, I am a scientific member of the HPTLC Association. The association promotes the use of HPTLC in plant analysis and other analytical fields. One of our members, Deborah, has developed the concept of comprehensive HPTLC fingerprinting during her PhD thesis. And today I would like to introduce you in the first part of our webinar to this concept, and in the second part Stefan will show you the technical solutions that CAMAG provides for HPTLC. Okay, in this first part of our webinar, I would like to start with the question, what is comprehensive HPTLC fingerprinting? The HPTLC fingerprint is well established for identification of plant materials, and it's included in pharmacopoeias worldwide. The HPTLC fingerprint is the image of the visual HPTLC chromatogram. Each track represents a sample. So the fingerprint includes application and front position and consists of a sequence of separated zones with defined positions and colors. Comprehensive HPTLC fingerprinting addresses also the overall intensity of the fingerprint and relative intensities of zones within. That means the electronic image of a sample, which is typically used for identification of herbals, contains also quantitative information. With this information, besides confirmation of the identity, right or wrong, the purity, mixture of different species or plant parts, and content of marker substances can be determined. So the example shows comprehensive HPTLC fingerprinting of Ginkgo products from a market study. You will see more details in the next few minutes. Identification and quantitative assessment are combined in a single low-cost test. The HPTLC fingerprint allows clear identification of Ginkgo products of good and bad quality. if compared to the fingerprint of a botanical reference material. And in this case, none of the samples meet the quality criteria as they all have an untypical pattern. On the first track is the reference material, which was spiked to contain the maximum allowed content of the flavonoid quercetin. And by a mouse click, the peak profiles from electronic images of chromatograms can be generated, and samples can be compared to a standard or a reference sample with a maximum allowed content. An easy limit test of multiple samples in parallel can be performed to determine compliance with the USP limit. In our market study, 26 samples have failed the limit test, and only 9 samples were within the acceptance criterion for quercetin, but failed because of other issues which we will see later on. So those have failed. Those were within the acceptance criterion for quercetin, but they also have failed because of other reasons. Let me briefly introduce you to the quality of herbals and botanicals and the current state of the regulations before we get back to the Ginkgo market study. What are botanicals and what are herbals? So botanicals the US uses the item or the name botanicals, and the Europeans are talking about herbals. They can be raw materials such as herbal drugs, powdered herbal drugs, fresh plants, and extracts, for example, tinctures and dry extracts, and products. Quality criteria and tests to be performed can therefore differ depending on the sample. For raw materials and extracts, we may find different monographs for identification because of the different sample composition. And to follow supply chains and to monitor processing, knowledge about the link between raw material and extract is needed. In a raw material and in the extract, or at the end, the herbal drug, several tests have to be performed for quality control. For the raw material, identification starts with botanical identity, established by macroscopic and microscopic investigation, and maybe also DNA barcoding, and confirms the name of a material. The chemical fingerprint is related to quality of the material for use as supplement, medicine, or cosmetic ingredient. And for extracts and finished products, DNA barcoding is usually difficult or not possible at all because of the chemical solvent present or used for extraction. The chemical fingerprint will still allow to distinguish different plant parts and to check for adulteration with related or other plant species. So, what about the regulatory situation? In December of 2015, the United States Pharmacopoeia has introduced a general chapter 203 on HPTLC. But before that time, the chapter 621 described the interchangeable use of either TLC or HPTLC. There was a lack of standardization as too many parameters were not well defined and described. In the new chapter 203, all parameters for HPTLC are defined, which leads to reproducible results if the described methodology is followed. So HPTLC 203 in a nutshell. The concept of standardized HPTLC is independent of instruments. The European Pharmacopoeia has a similar chapter, the chapter 2825, with the same parameters for a standardized methodology. What's in there? The standard plate for HPTLC is a 20 by 10 cm HPTLC glass plate silica gel 60 with a fluorescence indicator F254. The blade layout is defined as 15 tracks per blade with 8 mm band length and a distance from the left blade edge of 20 mm and from the bottom of the blade of 8 mm. Blades should be conditioned to a relative humidity of 33% prior to chromatography, and it is specified to develop blades up to 70 mm in a saturated chamber if nothing else is mentioned in an individual monograph. This will lead, or then you will get reproducible data every day and everywhere. And from a regulatory point of view, HPTLC is a concept that requires no special equipment. In this table, you can see all the standard parameters according to the General Chapter 203. And here our Ginkgo example by following a standardized methodology. So let's get back to the Ginkgo example. According to the USP monographs on Ginkgo leaves and Ginkgo extract, different tests have to be performed. For leaves, a total of 4 tests are done, and for extracts, 6 tests are done. And when we look at the raw materials, then we can see a chlorophyll zone in the upper part of the chromatogram and ginkgolic acid with different intensity. If there is a chlorophyll zone, then we know that this is a plant raw material and not an extract or finished product. The fingerprints can be compared side by side, and the pattern of each sample will show whether the characteristic zones for Ginkgo are present. Those are the fingerprints obtained with the Ginkgo method for flavonoids. After development, a derivatization with natural product reagent is performed, and an image of the blade is captured in long-wave UV light for fluorescence detection. If raw materials have been mixed with other species, the chemical fingerprint will show it. Here we can see the fingerprint of Ginkgo mixed with Sophora fruit, a known adulterant for Ginkgo. Additional green zones used as positive markers for adulteration are used as positive markers for adulteration with Sophora fruit. Let's have a look at the Ginkgo extracts. The fingerprint method is the same. And the fingerprints look similar, just with differences in the intensity of zones. We can still see that the origin was Ginkgo and that this is not just a synthetic mixture of flavonoids. Now we can check in this fingerprint, in addition to the identity, the content of rutin and quercetin. The total flavonoid content will not distinguish between individual flavonoids, but the fingerprint will show if an economically driven adulteration with other flavonoids has occurred. And the fingerprint of terpene lactones looks like these, but we will not discuss this further during this webinar. So how to generate data in HPTLC? First, we extract a sample or several samples. We apply them and develop the blade. Then we detect the blade as many times as needed to gain information about the sample composition. Within one analysis, the entire information is contained. The fingerprint can be detected prior and after derivatization in different detection modes. And this set of multiple data can be then used for evaluation. From each track in each detection mode, we can obtain a profile by scanning densitometry and/or from electronic images. So how to get the quantitative information from an image? A peak profile from an electronic image, or short PPI, has a start below the application position and an end above the solvent front position. So here start, there is the end. The application position is defined as RF value 0 and front position as RF 1. And the camera detects RGB values, and the software calculates the luminance in the middle of the band. For each track, peak profiles are obtained by a mouse click, and this will give us the information about the content of the different sample components or the content of a class of substances. The pattern of the entire sample is shown and telling the truth about its nature and origin. A marker approach can more easily be falsified by adding the target analytes for adulteration. The intensity ratios of the peak profiles within a chemical fingerprint will immediately detect cases of adulteration. So the strength of the HPTLC fingerprint is in the huge information obtained from each sample with one separation. The plate is an archive of separated substances because they are staying separated and bound to the stationary phase after chromatography. And this offers the possibility to detect many times in different illumination modes and with different excitation wavelengths. Furthermore, detection is possible with different detectors like a documentation system or a scanner in different stages of the plate, like prior and after chemical or biological reaction performed on the plate. Each time a detection is performed, a profile for identification and quantification is obtained, which can also be used for multivariate data analysis. So let's get back again to the Ginkgo example. We have one method and gain a lot of information out of one separation. After development, the plate can be excited with long-wave UV light for fluorescence detection. The native fluorescent zones of ginkgo and buckwheat, another known adulterant, are very different. Immediately, a case of fraud can be exposed. Then, prior to derivatization with the help of the fluorescent indicator in the stationary phase, and then exposed to UV254, UV-absorbing substances can be seen as dark quenching zones. An adulteration with Sophora fruit can be seen by intense zones at different RF positions compared to the quenching zones within the Ginkgo fingerprint. And to verify this, a second detection after derivatization can be taken into account. After derivatization of the sample with natural product reagent and exposed to long-wave UV light, adulteration with Sophora fruit will be seen by green fluorescent zones, not present in a Ginkgo fingerprint. After a subsequent derivatization with anisaldehyde, an adulteration with Sophora flower is visible in white light. So Ginkgo doesn't show those characteristic zones which are obtained within the Sophora flower fingerprint. So let's have a look at the market study and let's summarize the findings. 59 products have been investigated, and here you can see the results of the different products. On the first track is the system suitability test with chlorogenic acid and routine according to the USP monograph. And quercetin was additionally applied for the limit test. You can see that there is a lot of variation. A good Ginkgo fingerprint can be seen in the Ginkgo product sample number 20 and the products 36 to 43. So those are here. Only 11 of the investigated 59 Ginkgo biloba products complied with specifications of the USP for identity, limits on routine and quercetin, and content of total flavonoids. 8 products lacked zones characteristic of Ginkgo and showed mainly a zone due to routine. For example, the Ginkgo product number 14. There is a very intense zone of routine, but the pattern typical for Ginkgo cannot be seen at all. One sample contained only 5-hydroxytryptophan, which is this one. And one sample showed additional blue zones between RF 0.6 and 0.7. 16 samples showed additional green zones between RF 0.2 and 0.3. So for example, number 3 and 4 and the number 17. And most of the time combined with high levels of quercetin. Based on the identification results, it was concluded that many of the analyzed products seem to have quality problems, including adulteration. And in addition, the intensity of some fingerprints was lower than expected. So the purity can be checked by different detection modes used on the same plate by comparison with the fingerprints of the known adulterants. In a mixture, the presence of the other plant species down to 10% can be detected. So this is a typical fingerprint of Ginkgo biloba leaves, this one of Sophora fruit, this one of Sophora flower, and this one of buckwheat herb. And those are mixtures with the typical adulterants down to 10% with Ginkgo biloba. For limit tests, the specified quantity of the marker compound is applied. So here for routine and here for the quercetin. And the intensities measured at different detection modes are compared. Here in the shown graphs, 3 samples have failed the limit test for routine. So they are above this line. And here, 3 samples have failed the test for quercetin. In general, most of the products that were compliant with the total flavonoids assay had weak fingerprints by HPTLC, but intense zones at the positions of quercetin and or routine or additional zones. To properly detect addition of routine and quercetin, an additional HPLC limit test is required for extracts by USP, and however, the combination of HPLC assay and HPLC limit test and the limit tests performed by HPTLC in combination with identification have given the same results. So I would like to point out again, all those HPTLC results for each sample were obtained from one separation just by multiple detections on the same plate. Let's have a look at another topic, at another concept, and first have a look at system suitability test according to the USP. A system suitability test in HPTLC or in general shall verify that the analytical method was suitable for its intended purpose when the analysis was performed. The USP uses the SST to check the suitability of the system for resolution, position, and color of the bands, and consists of 2 or more reference substances that have similar but just separable RF values under the chromatographic conditions to be used. Here is an example. So there is the SST with 2 barely separable substances. And it's just focusing on a specific region of the chromatogram. If they are seen as 2 zones, the SST is passed. procurement of chemical reference substances required for these SST may add significant cost to routine analysis if laboratories are applying many different HPTLC methods. And they might be time-consuming if laboratories have to develop and validate different sets of SSTs. This is why I would like to show you another new concept for HPTLC here. And this is the new Universal HPTLC Mix, or short UHM, for use as general SST. The introduction of a universal HPTLC mix usable with many different developing solvents of different polarity. So for the development of a universal HPTLC mix, in a first step, 24 organic substances were tested with 20 different developing solvents. And at the end, a mixture of 8 substances was selected. It is suitable to be used with different developing solvents. At least 3 to 4 substances can always be seen in UV254 with a distribution over the entire chromatogram. Then in the second step, the capacity of the UHM to detect changes in the chromatographic conditions was investigated. Changes in the relative humidity was good. Changes in the composition of the developing solvents could be detected. And changes in the chamber configuration with or without saturation were also detectable. Then in the third step, intra- and interlaboratory trials with 5 HPTLC equipped labs were performed. The confidence interval within the same lab was within an RF shift of 0.03 and within different labs of 0.04. The concept was published by Ginkgo and co-authors in the Journal of Chromatography A this year, and the UHM as a product was recently launched by Sigma Aldrich, a subsidiary of Merck KGaA. And how can this be used from a practical point of view? So we can apply the UHM on the plate together with standards and samples and define the zones detectable in the UHM in UV254 after chromatography. For each defined zone, we obtain a certain RF value. If we run this now several times, for example 3 times the same day and 1 plate on 3 different days, we can set our acceptance criterion, the delta RF values. And within a method document, we can then specify our RF with the acceptable tolerance, and then for each analysis we can check if the chromatographic conditions were okay by checking if the RF values of the zones obtained by the UHM are within the tolerance window. So we define our SST, we label here the zones, then we get RF values during method development and also the delta RF value. And then when we run an analysis, it will check if within these windows there are zones detectable from the UHM. So we have an automated check. And then we get pass or fail. So at the moment, a UHM is not a requirement in any regulation, but it can help to increase the reproducibility in HPTLC and to improve the comparability between different labs. Furthermore, it can be used during own method development instead of searching for suitable phytochemical reference substances. And now I would like to talk about how to interpret an HPTLC fingerprint in accordance with regulations. Here is an example monograph for the identification of ginkgo leaves and ginkgo extracts according to the European Pharmacopoeia. The general chapter 2825 introduced the concept of intensity markers to semi-quantitatively interpret an HPTLC chromatogram. Our result table gives the information about the zones that must be detected in a sample and their color and intensities, faint or faint to equivalent, in comparison to the intensity of the markers. With the help of a suitable software, profiles from an electronic image can be used to translate this table and to assign it to the zones. So here's the fingerprint, here's the profile, and here is the assignment to the zones that should be seen according to the table. We have seen before how image profiles are generated. And now a next step for manufacturers could be to define quality criteria of their products based on a representative HPTLC fingerprint obtained by merged fingerprints of multiple samples of good quality, representing also the natural variability and the range of the intensity of the characteristic zones. If a raw material or extract is within the range, it is a pass, good sample. If it's not, the sample has failed. So let's do a check and see if all are within the acceptance criterion. And now with this animation here, I would like to end up with my part. We have one sample separated and get profiles from images or scanning densitometry in different detection modes prior and after derivatization. This means that we can define unique quality criteria for each botanical during method development and use comprehensive HPTLC fingerprinting to check the quality of complex samples. One sample and multiple detection modes. And now with this, I would like to hand over to Stefan for the second part of our webinar.
Thank you, Melanie, for presenting this interesting concept of comprehensive fingerprinting. I would like to point out that this is based on the research outcome of scientists within the increasing HPTLC community. In this second part of the webinar, I would like to show you CAMAG's technical solutions for performing HPTLC. The company CAMAG was founded in 1958, starting with the production of aluminium oxide. Soon, we began to engineer and manufacture instruments for thin layer chromatography. In the 1980s, we added software to control the automation of the TLC steps and to evaluate the results. This was the birth of high-performance thin layer chromatography, or short HPTLC, where each step is standardized and automated. Recently, we went one step further with the introduction of HPTLC Pro. This new system features a modular and fully automated workflow throughout all processes, which meets the standards and requirements of the modern laboratory. To meet your standards as precisely as possible, we created 3 segments that we provide instrumentation, methods, and support. First, the conventional TLC that is used in thousands of laboratories around the world. TLC provides qualitative results, for example, to monitor processes during chemical synthesis. Second, there is the field of HPTLC. With this technology, you achieve reproducible qualitative and quantitative results. HPTLC is suited for small companies and research labs to analyze their samples. Third, if you want to process an increased number of samples in a standardized, fully automated workflow, we offer the newly developed HPTLC Pro. Working with this system requires only minimal manual intervention, and it is therefore optimally suited for quality control laboratories in mid-sized and large companies. All CAMAG HPTLC instruments, including HPTLC Pro systems, are controlled by the same CAMAG software, called VisionCats. The VisionCats software allows you to work with standardized methods and to get traceable process parameters and results throughout the whole analytical process. As many of us work in regulated environments, such as under GMP or GLP, we added the 21 CFR Part 11 option to ensure data security, safety, and integrity, but also user management, audit log, and the use of electronic signatures. VisionCats contains a method library of validated instrumental methods for all licensed users. Currently, there are over 200 methods in the field of botanicals alone, and those are available for download. VisionCats allows you to set up individual methods on the natural user interface. You can add instruments from all segments, including TLC, HPTLC, and HPTLC Pro, by simply drag and drop the steps to the workflow. After analysis, you can generate linear profiles from images or record them with scanning densitometry. Both identification and quantification can be performed for the analysis. For example, we can prove the identity and purity of a substance by comparing the UV spectrum of a sample on the plate to a reference spectrum from another track, run or from the comparison files available from the method library. Another option is to generate a calibration curve and calculate the content of an unknown sample via its peak area. We are constantly expanding the method library with ready-to-use methods. The VisionCats software features the comparison file option, which allows to compare sample images with reference images. For all methods, we include the SOP and method documentation as well. Such an example is the identity check of the plant Simicifuga. Track 1 and 2 is our system suitability test, the SST. When the SST is within a certain range of RF value, we can compare substances analyzed on different plates. With VisionCats, you can compare, rearrange, and group different samples during data interpretation process to create a virtual plate. Related plant species can be distinguished by the HPTLC fingerprint. A typical HPTLC workflow consists of the following steps. Application, where we typically spray an 8 mm band of sample onto the plate. Development, performing the chromatographic separation in a standardized environment with controlled humidity and optionally conditioning. In the derivatization step, the derivatization reagent is sprayed as a fine mist of microdroplets into a chamber and allowed to settle onto the plate. This guarantees a homogeneous application of the derivatization reagent. The process requires significantly less reagents than other alternative derivatization methods, and it is safer in handling for the user. To evaluate the separation and to generate peak profiles, the TLC visualizer and TLC scanner are best suited. If required, HPTLC can easily be coupled to mass spectrometry by the MS interface. Bringing it all together, the new HPTLC Pro is a modular system that transports the plate autonomously between the modules. There is much more about this new line, beside the automation, but this is beyond the scope of this webinar. Several videos and webinars of HPTLC Pro can be found on our webpage. To get deeper into the world of HPTLC, we have been creating several tools to explain the technology and its applications. On our webpage, camag.com, you can find all instruments, methods and educational material. Twice a year we publish the CBS magazine, where we present application examples from other HPTLC users from around the world in various fields of application such as botanicals, food and feed, forensics, pharmaceuticals, cosmetics, environmental and biotechnology. More than 11,000 abstracts on TLC and HPTLC applications are available free of charge in the section CCBS database on our webpage. Each abstract gives information on the methodology, including stationary phase, developing solvent, detection, and so on. Please also see our videos about instruments and applications and connect with us on social media. So let me conclude. The HPTLC fingerprint allows to identify a substance or a group of substances in a complex matrix and to compare the spectrum with other samples or references between different plates. The information acquired goes beyond the HPTLC fingerprint, which allows quantification of the substances and further detection steps, including derivatization, with just one single instrumental setup. The sample preparation for HPTLC is quick and simple. Because the stationary phase, the HPTLC plate, is only used once. Almost any matrix can be directly applied onto the plate, which reduces sample preparation time and the use of additional consumables, and therefore saves substantial money on the sample preparation process. For large sample batches, HPTLC Pro automates the entire HPTLC process. At CAMAG, we put the HPTLC user experience in the center of what we do. It means so much to us to make a difference in people's laboratory experience. The future of HPTLC is automated, simpler, and more convenient.